Welcome to the course on qPCR Diagnostic Mastery, qPCR as a molecular diagnostic tool. The learning objectives are understanding the basic principles and applications of qPCR in molecular diagnostics, interpret qPCR data and assess its limitations, design and validate qPCR assays for specific targets, and apply qPCR knowledge to various diagnostic scenarios. So this course is designed to be in 4 modules. 4 modules are Introduction to qPCR, Design and Optimization of qPCR assays, Advanced qPCR techniques, and case studies and applications of the qPCR. So in this module 4, we will take certain cases and design assays and implement these your knowledge in designing the assays and how to optimize and validate the assay conditions. So these are the 4 modules. Today I will be covering module 1, which involves the basics to the qPCR and the applications of qPCR, and overview of what PCR is and what are the applications and how we can interpret the qPCR data. And in coming modules, we will delve deep into the primer probe, design consideration, and assay optimization conditions, and various advanced qPCR techniques. And in module 4, we will take some of the assays and do how to design the primers and probes, and how we can optimize these parameters. So these are the 4 modules which we will be covering in this course. So let's jump directly to the basics of qPCR. So what is qPCR, or in fact what is PCR? PCR is polymerase chain reaction. It's a molecular biology technique that used to amplify and analyze the DNA, and it involves 3 major steps, that is denaturation, annealing, and extension. So the 3 phases of the PCR cycle, the denaturation, annealing, and extension. In denaturation phase, the double-stranded DNA gets separated into single-stranded DNA, and during the annealing phase, the primer present in the master mix solution will anneal to the separated DNA strands, and in the extension phase or the elongation phase, the DNA strand get extended. The primer attached to the DNA strand get extended by the polymerase that is used in the master mix. So DNTPs will get incorporated into the primers, and this gets extended. So the denaturation typically done at a higher temperature, that is around 94 to 95 degree centigrade, for about 1 or 2 minutes for the initial denaturation, and during the cycle it is kept as 10 to 20 seconds in real-time PCR conditions. And once the denaturation is done, the next phase is the annealing of the primers. So in this step, the temperature is reduced from 94 to the primer annealing temperature. So typically that is designed based on your requirements and the TM of the primers. Typically the annealing temperature is set 5 degree below the TM value of the primers. The primers are short oligonucleotides of 20 to 30 base pair in length. These are specific sequences that are targeting to your DNA template. So once that is done, so we keep the annealing for around 30 to 34 seconds, wherein the primer binds to the DNA strand in a specific region, and followed by increasing the temperature to 72 degree. That is optimal temperature for Taq polymerase. During that phase, the attached primers get extended by the Taq polymerase, and the nucleotides get incorporated into the extension phase. And at the end of these 3 processes, you will get double the amount of DNA that is present from the initial. So 1 strand become 2 strands, and it can be expressed by the general formula 2 to the power n. So n represents the number of cycles, and 2 is the doubling of the DNA strand. So when you have initial starting copy of 100 molecules of DNA, so at the end of 40 cycles, you will have 100 into 2 to the power 40 copies of DNA that is present. So PCR is a very sensitive technique, and that copies the DNA into billions of copies starting from very low number. So that is the basics of PCR, and now we can look into the differences between qPCR and conventional PCR. So there are 2 types of PCR. One is conventional or traditional PCR. The other one is the qPCR. qPCR is a real-time PCR. It provides real-time monitoring of the amplification process It allows for the quantification of DNA during each cycle. It utilizes the fluorescence probe or dyes for detection, and it enables the determination of starting amount of DNA in the sample. So qPCR is basically a real-time PCR, or it monitors the amplification in real-time process. And the advantage of this qPCR is it allows for the quantification. So the bacterial or viral load of the sample can be determined using the quantitative PCR, and the method used is fluorescent probes or dyes for the detection. Whereas in conventional PCR, it is also known as the endpoint PCR or endpoint analysis, where amplification is measured after the reaction is complete. So there is no real-time amplification monitoring in conventional PCR, and it does not provide real-time data during the reaction. We will have to wait till the reaction is completed, and the detection is usually based on the gel electrophoresis. You will have to run the PCR product onto a gel to visualize whether the sample has amplified or not. So these are the basic differences between qPCR and the conventional PCR. In qPCR, the fluorescent detection and quantification can be done using either fluorescent dyes or using probes that emit signal when bound to the amplified DNA. The fluorescence is measured in real time during each PCR cycle, allowing the continuous monitoring of DNA amplification. So there are two types of strategy that can be used in the real-time monitoring. One is the SYBR based dye-based assay, and the other one is probe-based chemistry. So SYBR is a intercalating dye that binds to any double-stranded DNA. So during the amplification, as in when the doubling of the DNA occurs, the more and more amount of the dye that is in the master mix will get bound to the DNA, and that can directly give correlation between the amount of DNA that is produced and the fluorescent signal. So once the SYBR bounds the DNA, it emits fluorescence. Without binding to the DNA, it is having very minimal fluorescent signal. But when it binds to the double-stranded DNA, it will start emitting a relative amount of signal. So that can be captured. So the amplification signal increases as the amplification of the DNA in the sample proceeds to higher level. So the fluorescence also increases accordingly. Whereas in Taqman chemistry, or the Taqman probe-based chemistry, probe is a short stretch of oligonucleotide having a reporter and a quencher. The probe is very specific to the target region of the template, and it binds next after to the primer region. So during the elongation phase, the Taq polymerase extends the primers, during which it encounters probe during the amplification phase. Due to the exonuclease activity of Taq polymerase, the Taq has the ability to chop off or cleave the probe that is present in the middle of the elongation phase, and that results in the cleavage of probes from the DNA strand, which is bound during the annealing phase or during the extension phase. So what happens is normally a probe that is in its native phase will the probe reporter and the quencher will be in close proximity, and the fluorescent signal emitted by the fluorophore or the reporter will be captured by the quencher. But during the chopping phase or the elongation phase, the Taq polymerase cleaves the probe, and that makes the reporter and quencher far apart, and there will not be any quenching during that phase because it is further apart from the reporter. The quencher will be further apart from the reporter. So the fluorescence emitted will not be quenched by the probe, the quencher, and you will get a fluorescent signal. When the DNA amount present in the sample increases, your fluorescent signal also tend to increase accordingly. So that is a basic principle between probe-based chemistry and SYBR based chemistry. The SYBR binds to any double-stranded DNA emitting fluorescence, which is simple and cost-effective, but it requires a careful primer design because it can bind to any double-stranded DNA. So even the dimer can get detected during this phase. So that can lead to a false result. So careful primer design consideration is required for SYBR based chemistry. Whereas Taqman based probe utilizes a specific fluorescent probe that is cleaved during the DNA synthesis or the amplification phase. This is very specific and provides very sensitive detection technique or the strategy for this qPCR. The advantage of Taqman probe is that it can be used to detect multiple pathogens in a single tube, so multiplexing is possible, whereas SYBR green, there is a limitation of using SYBR green for the PCR for detecting multiple targets in a single reaction. There are ways to do that by using a melt curve based analysis, but Taqman probes based chemistry is more popular and advantageous in case of doing a multiplex PCR. Coming to the various applications of qPCR in molecular diagnostics, we can see that the qPCR is widely used in detection of infectious diseases. During the COVID, this has taken into an exponential level. The applications of qPCR has been widely utilized for monitoring and detection of virus and pathogens. The advantage is that qPCR can give quantitative information about the pathogen that is present in the sample, whether it is a virus or it is a bacteria, and any form of pathogen that is present can be quantitated. That means that the amount of the bacteria or virus present in the sample during an infection can be identified. And we can also use this for cancer diagnostics to monitor certain mutation and also to identify the gene expression patterns and monitoring of minimal residual disease, meaning that during the cancer phase, if you are giving a therapy or a drug to treat that mutation, the amount of disease that is present can be monitored by monitoring the gene expression levels. So when you give a medicine to a patient, the levels of certain proteins and the RNAs, so RNA tend to release proteins. So the level of mRNA can be analyzed, whether the very upregulation or downregulation, and the effect of treatment can be monitored using qPCR. One of the classical example is the use of qPCR for monitoring the chronic myeloid leukemia, where the BCR-ABL transcript can be monitored during the course of therapy and provides information on the reduction of disease burden or the mutation burden in the patients. The other application involves the qPCR in genetic testing. This is very useful for premarital screening and to identify certain genotypes, and also it is used for providing personalized therapies. So certain individuals will have certain mutation SNPs in their genetic makeup, and that can cause adverse effect for certain medications. Certain medication may not be suitable for people with certain mutations, so that can be identified, and the medicines can be tailor-made accordingly. Other application involves qPCR in forensics, meaning to identify individuals based on their unique DNA profiles. So there are advantages and disadvantages for qPCR. The advantages include high sensitivity and specificity. Quantitative information can be available in real time. You don't have to do any gel or any other kind of post PCR modifications or techniques to identify. It can be obtained during the amplification phase itself. The multiplexing capability of detecting multiple targets is very useful, because even with a single PCR run, we can detect multiple targets. The disadvantages include cost and complexity of the equipment, because it requires qPCR instrument which is bulky, and the cost of running those instruments are expensive. It require master mix and primers and probes designed for that. In fact, it is very sensitive, but at the same time, it is cost wise it is high compared to the rapid test like lateral flow test. So compared to that, qPCR is expensive. qPCR is susceptible to inhibitors from complex samples like sputum and other body fluids. It may be difficult at times to have a very good pure sample, so you need to have a strict or well-versed nucleic acid extraction step upfront for the PCR to perform well. So these are some of the disadvantages and advantages of PCR. Coming to the amplification, if you are analyzing the data from PCR, the first thing is the amplification plot. So amplification plot is typically a sigmoid curve, attains a sigmoid curve. It has exponential phase and non-exponential phase, and initially there is a lag phase. We can say that during that phase you will not see much of an amplification. It tend to go in the baseline, but at certain point it starts to exponentially amplify. So that is the detection threshold. So after a certain threshold, the signal start to give exponentially. So that is where we call this phase as exponential phase, and the CT value is the value that represents cycle number at which the fluorescent signal crosses the predetermined threshold. So till then it is not detected. So after the threshold value, we can see an exponential amplification. So that is called as the cycle threshold. Certain manufacturers use this as CT, certain manufacturers use this as a CQ value, some use as a CP or the crossing point. So all these are relating to the same concept of crossing the threshold value of the background signal. So the qPCR is very informative, and it provide information of the target pathogen that is present in the sample. So this can be achieved by running a standard curve of non-concentration. It can be plasmid or any other standards. Typically, standard curve is done by serially diluting the sample of non-concentration and generating the amplification curves and identifying the CT value. And when you plot these CT values with the copy numbers or the concentration of DNA that is used for the PCR, you get a straight line, and from where we can calculate the slope and intercept value, and also we can calculate the PCR efficiency. So based on these inputs or information, we can calculate the bacterial load or viral load of an unknown sample. So that is the use of standard curve in the analysis. A standard curve is very important. That can give or useful at times for calculating the quantity of unknown sample, and it serves as a reference for determining the initial amount of DNA and RNA. So based on this, we can calculate based on the slope and intercept value. We can calculate the viral load or bacterial load of an unknown sample. In certain cases, we'll have to normalize the qPCR data. That's because it compensates for variations in the sample quantity and quality. And these methods include use of reference genes, internal controls, or normalization to the nucleic acid content. So we can normalize to these ratios based on the reference gene expression or presence of internal control, and it is useful in normalizing the content. Hope you are clear with the module 1. If you have any doubt, please feel free to drop comments in the comment section. I will answer those. And in the next session, we will learn more advanced techniques. So this is just basic introduction to the qPCR. I will be covering much more depth of information in the upcoming modules. Thank you.